Okay, good afternoon, and first of all, thank you very much to the organizer for giving us the opportunity to come here and talk to you about our project, our work. I am a postdoc. I am working on data mining issues, let's say, and as a part of the air microbiome project at the CELSI Research Institute, which is located at NTU, by the shared research institute between NTU and NUS. And I'm going to give you today an overview, a vision over our project, which started approximately 2 years ago. Here in this slide, you can see three of the major components of the Earth biosphere associated with the estimated amount of biomass that can be found in them. And just to make the point that the air, despite being the largest component of the biosphere by volume, it has a far lower, very low, let's say, concentration of cells, specifically bacterial cells, as compared to the soil, for example, or to the ocean. And also that the air, despite being so important for our health, despite having such a big impact on our health, you just need to think that each day we inhale approximately 10 to the 6 bacterial cells, and for sure some of them, maybe some of them they have no effect, but for sure some of them they have some sort of effect on our immune system or on our health in general. Despite all of this, the air is an ecosystem that is, at the day of today, still largely unexplored, let's say. However, there is an increasing interest of the international community on the study of the air microbiome. Here you can see a few key papers, a few key studies on different types of air microbiome that have been performed around the world in the last, before 2013. And here you can see that also in the very last 2 years, 2014, 2015, still many studies are starting to appear here and there around the world. But maybe what's more interesting from our point of view is that in this area, the tropical area where we are, there is not much research going on at the moment, which makes our study on the air microbiome of Singapore possibly even more, let's say, relevant. Also because in Singapore we have a great variety of different environment. Well, after the presentation of this morning, I don't know if we can say that in Singapore we have forests, but they say that we have something which is similar to forest. We have commercial type of environment, industrial, residential. We are surrounded by the ocean, but we also have waterways across the island, which makes Singapore a very, very good case study. What you can find in the air in terms of biotic particles can be either fungi, bacteria, viruses, and apart from the viruses that we can say that remain more or less permanently suspended in the air, everything else, bacteria, fungi, they all have a specific deposition time, settling time, which means that in absence of perturbation, after a certain amount of time, they will just settle. That's why we are very interested in studying all those sources and sinks, for example, in the outdoor and in the indoor air, because so everything that we can find in the air, it has to come from somewhere. So if we are outside, for example, it may be something that has been suspended or resuspended from the soil. It may be something that is coming from the plant, or maybe something that has been aerosolized from the ocean. Or the same way, if we are indoor, we also can have sources or sinks of bacteria. If we are indoor, also the human being is an important contributor. For example, right now I'm speaking to you, and I am emitting some bacterial cells in the air. If I do this on my skin, I am releasing, aerosolizing some skin cells together with the associated bacteria, and so on. And in a similar way, the air conditioning system that is working in this room right now is working the opposite way, filtering, so trapping the particles on it. so acting as a sink. And we are interested in all those kind of things. In the last 2 years, we have been working on establishing what we can call a solid analysis pipeline. And I must say that has been quite a challenging task, because as I mentioned before, the amount of biomass that you can capture, that you can sample from the air is extremely low, which means that we are working very close to the level of the noise, to the background noise. In other words, basically, if you think about it, we are trying to sample, to study the same type of environment in which we work. We collect an air sample, but every action that we perform on the sample is also performed in the air. So we are very much exposed to the risk of contamination and picking up false positive at any point of this analysis pipeline. So I'm gonna guide you very briefly through what is we can consider a typical analysis pipeline for us. We use these air sample devices that you can imagine them as like some very powerful vacuums. They're not vacuums, but you can imagine like that. They suck air at very high speed, high volume. They suck air in, and the particles, they get trapped either on a filter, if it's a filter-based air sample, or on a liquid, if it's a liquid-based one. And then after that, after we collect this sample, we move to the lab where we have to perform a DNA or RNA extraction task, typically DNA, which means that we also had to come up to optimize our protocol to work on very low biomass samples, okay? Whatever we get from the DNA extraction, we take this DNA and we proceed to what we call metagenomic sequencing. So for the people who are not familiar with the term, you can imagine metagenomic sequencing like, so if I have a sample, let's say an air sample that I know that is full of 1,000 or million of microorganisms, if I perform a metagenomic sequencing, it means that I go and try to read all the DNA fragments that are in my sample, okay? And of course, also here, also in this step, I am exposed to to a lot of risk of, you need to be very careful. You may end up sequencing the noise rather than the signal. You have to know how low you can go with the biomass, et cetera, But let's say that you get a good sampling, a good sequencing result. Then you have million of sequences, let's say, and you can move to the very last part of the analysis pipeline, which is the data analysis or bioinformatic analysis, where I now have all these sequences. I just don't know what they are. So this step we call taxonomical identification, because we need to understand what these sequences that we have in our sample are. And this step is typically performed by comparing all our sequences against very big databases where that contains the genome, or full genome, or partial genome, or fraction of the genome of all the organisms that have been studied or sequenced until now, which is also a very computationally expensive step to perform. So take a look now at this slide. You can see here 75 air samples that have been sequenced. On the y-axis, you can see the number of sequences for each sample, so 2 million sequences for each sample. And we tried, using those very big databases that I just mentioned, try to identify what was inside our samples. And that gray part of the bar chart that you can see on the top is approximately 50% of the sample. Those are all what we call no hits, which means that there is nothing in the known databases, in these big databases, which is they're not even close to be similar to anything which is known up to the day of today. So we have absolutely no idea what those organisms are, okay? The other part of the bar chart, the pink one that you see there, approximately 20–25% of the sample. For them, we do have a hit here, not assigned. We call them not assigned because we do have a hit against the database, but we are just not very confident about this. There is a hit, an alignment, a matching, but it's not a very good quality matching. So according to the parameter that we want to maintain to have a certain level of confidence on our matching, we prefer not to make any decision on what this sequence belongs to. Could be, could not be, we prefer not to, we're not confident enough. And only, you can see 10 to 50%, the light blue and the yellow portion, eukaryotes and bacteria, 10-50% only is the portion of our sample that we are able to identify. Here you can see only high-level bacteria or eukaryotes, but we are able to identify them up to the species level. And these samples, this plot looks like this because we are talking about air samples. If those samples were instead, for example, fecal sample or marine sample, something that has been studied very deeply in the last 100 years, then I'm not an expert of marine sample, but we can guess that maybe 80-90% of those sample we would have been able to identify what was there because they have been studied for many, many years. But because it's hair, we don't know much. That is why, on top of using this approach, this comparison with very big databases, we are also building our own database of complete genome assemblies isolated from the air. So what typically happens when you try to sample the air using the air sample that I showed before, you don't get enough biomass to build a full genome, though. So you try to isolate, to culture them, certain organisms in the lab. You let them grow. When you have enough biomass, using a combination of PEC biotechnology and Illumina HiSeq shotgun sequencing, so a double validation system, we are able to confidently close the genome. The technical term is assembly the genome, which means that reconstruct the entire genome of the organism that we were growing in the lab. And up to the day of today, we managed to assemble the full genomes of 50 fungal species and 50 bacterial species. Of course, this is not enough, but it is something that we use on top of the traditional methods to enhance our analysis. And we are also getting ready to release this public database. We came up with this funny name, DNAir, where we're going to put all these full genomes. We're going to provide a lot of information what these organisms are, where they were found before, etc., etc. On top of that, we also use microscopy. This is a compilation of organisms that are found in the different samples. The tiny one, very tiny one, are most likely bacteria, while the slightly bigger one are eukaryotes, so most likely fungi. And this big chunk that you can see there is what we think is a portion of an insect, so most likely the leg of a fly, for example. Just to give you a visual reference of what an air sample looks like. Now, take a look at the result of this experiment that we performed in the last weeks. We call it day variation. We wanted to assess if the microbial composition of the air varies across different moments of the day. So we performed this from Monday to Friday, 5 days. See there at the top. We started sampling from 5 a.m. in the morning to 7 p.m. in the afternoon in a time point of the duration of 2 hours, because 2 hours is the amount of time that we need to run the air sample for in order to get enough biomass for sequencing. So the first time point is from 5 to 7, then we take a break, and then we have 9 to 11, take a break, 12 to 2, etc., etc. 5 time points per day, and each of them has 3 replicates, okay? And the size of the bubble that you see here is proportional to the number of sequences that have been assigned to that specific microbial species. Okay, so the bigger is the bubble, the more abundant was that species in the sample. And I would like you to note this very high level of, let's say, reproducibility of this result. We are quite confident that this level of robustness, let's say, haven't been really achieved before by other labs working on the same type of problems. I mean, being able to detect such small variations, such as you can see there at the top, some bacterial species showing up at a specific moment of the day and then disappearing at other specific moment of the day. So this is something that we are very, let's say, happy or proud of. We have failed 1 million times, but we managed to come out with this solid and reproducible result. That's why we're getting ready also to publish our protocol, because we would like to establish a standard for the analysis of air samples. If I had to spend a couple of words just on the meaning of this experiment per se, then I would say that, as I mentioned, you can see that we have discovered that there are specific species that they show up and disappear at a specific moment of the day if we perform more analysis. But if I had to show just one more slide on this experiment, I would show you this multidimensional scaling plot, which shows you that all the early morning sample, for example, they cluster. They are very different from any other sample in terms of microbial composition. And then if you go on through the day, you can see here, if you move 9 to 11, the red one, you start to be slightly different. And then you go a bit around noon, the green ones, you are here. And then after noon, you slowly start to come back around 3 to 5, you're here in pink, and then you're here at the end, light blue, again, in the evening sample, being much more, let's say, similar to the morning sample, indicating that there might be I would say dial cycle in the microbial composition of the air. That's why we already repeated this experiment, but now we included also the night in our sampling schedule, and we don't have any more gaps in between time points, so we sample around the clock for 5 days, night and day. And we are about to get the result of this one as well. Also ongoing is on the same fashion as the day variation. Now we also have vertical variation and horizontal variation, which means this is to establish, for example, if I take an air sample here at the height of my nose, is this representative of what? If I go 2 meters more high, it's going to be the same or it's going to be different? If I go 10, if I go 20, how does it change? And the same thing in horizontal. Because our, let's say, ultimate goal is to come up with a Highland-wide characterization of the microbial composition of the air or in all areas of Singapore, so that we can say in certain area we have some specific, for example, bacterial taxa that are more abundant, and why. But most importantly, I would say that we want this information to be useful to improve the quality of life in Singapore or also maybe somewhere else. In fact, we are already collaborating with immunologists, expert in allergies, expert in specific respiratory tract diseases such as asthma, in order to find a link between what we find in the air and those type of diseases. Okay. This was my vision, let's say, over the Air Microbiome Project, which, of course, is a joint effort of many, many people. Thank you, and if you have any question, we'll be happy to answer.
I guess that with food we also get a lot of bacteria into our body. Are they very different from the ones in the air, or are they the same types and the air is much less?
What do you say, sorry? If
with food, we get a lot of bacteria into our body.
Yeah.
Are they the same type of bacteria, and if so,
you mean if the bacteria that we find in the air are this? No. So the list that I showed before of the day variation experiment, for example, we show this list. In that slide, you only have the top 40, but there are many, many others. We show this list to some, let's say, expert microbiologists, and they say that they cannot recognize almost, they know almost none of the species that showed up in that sample, which means that are not particularly well known.
I have a question. What fraction of the eukaryotic material is pollen? Tree pollen, crop plants, and so forth?
A big part of it, more than 50%, I would say.
Okay, thanks.
Any more questions? Okay. Just curious whether anybody has attempted some kind of a transport model.
Not yet. We are not there yet, in the sense that transport model is, in order to take one one sample, we need to run the machine, the sample for 2 hours. And we are aware that during this amount of time, also the air, maybe because of the effect of the wind as well, is moving. But we're not able to capture the movement of the air to this type of resolution yet. Because if we sample for less amount of time, then we don't get enough biomass.
Hi, just wondering, are there any plans to characterize the indoor air microbiome and perhaps compare it to the outdoor and what happens when the
Yes, also the indoor, but the problem that we are facing with the indoor spaces is that we need so much air to sample, to suck in, to sample so much air in order to get enough DNA or biomass for the analysis, that any indoor environment at the moment would be too small to be sampled without just running out of air. Unfortunately, it's true.
Probably is a perfect time to close the session. Thanks to all the speakers. So.